Hello everybody. Welcome to the Antibody Society webcast on antibody validation. In this, the fifth episode of the series, we're going to start examining practical issues, beginning with the fundamental issue of using appropriate positive and negative controls. As usual with antibodies, this is not as easy as it sounds. The Antibody Society is an international non-profit association. We're an international forum for facilitating discussion and disseminating information, providing guidance and assisting in training and advancement of students and postdoctoral fellows. If you care about antibodies, please join the Society. The speakers in this webcast, experts from academia and industry, are expressing their independent assessments and are not representatives of the Society. Viewers can write in questions at any time for the speakers to answer, and they will remain online for 15 minutes after the cast has ended to answer questions. So far, we've seen that if you're not very cautious, commercial tool antibodies can seriously mess up your science. For example, they may not be what's written on the label. They may not be made and characterized by the companies you buy them from. The accompanying datasheet may not refer to the reagent you have in the vial in front of you. And if all of that is okay, you may not have yet selected the best available antibody for your experimental context. Presuming that you've managed to avoid these obstacles, you can happily get on with your experiments, which will be the theme of the next casts in this series. But the first essential is that you must decide if the binding of your antibody is valid, and for that you need the appropriate controls. In this, the fifth episode, I'm very happy to be able to welcome Professor Giovanna Roncador from CNIO in Madrid. Giovanna established the European Monoclonal Antibody Network, and she's the chairman of that network now, and runs a team who routinely make and characterize very many monoclonal antibodies. Greetings, Giovanna. Thank you very much for joining us today. Giovanna, I'm sure many viewers think that obtaining and using appropriate controls for antibodies is straightforward, but that's a mistake. Please could you explain the issues and how users can make the best out of their control reagents?
Yes, thank you, Simon, and hi everybody, and welcome to the Antibody Validation webinar. What I would like to talk to you today is about the importance of the use of appropriate positive and negative control during antibody validation. First of all, it is important to define what is the meaning of antibody validation. We consider that an antibody is validated when we can demonstrate that it is specific, so able to bind to a unique epitope, that is a small fragment of a protein of 5 and 6 amino acids. Protein can be highly homologous, and they could, in some cases, contain the same epitope. So we have to be sure that our antibody reacts only with one antigen and do not cross-react. The antibody has to be selective. Experiment has to be repeated both over the time and by other researchers, giving the same results. Has to be reproducible. It is important to understand that since antibody can bind to linear epitope, linear sequence, or conformational epitope, three-dimensional shape, they can perform well in one technique, for example, Western blotting, and not in other, like flow cytometry. For this, validation must be carried out in an application-specific manner. Things get further complicated when we use techniques that use fixatives that are essential to maintain tissue architecture, like immunohistochemistry or immunofluorescence. Thus, an antibody that recognizes an epitope conformational in fresh tissue could not do it in fixed tissue. The reason for this is that formalin fixation produces methylene bridges between free amino acid groups and other functional groups of protein, resulting in conformational changes. In this way, an epitope that was accessible before is now masked. These changes can be reversed using antigen retrieval that use high temperature heating specific buffers. Fixation can also create cross-linking between tissue proteins, creating a sort of a net that masks the epitope, so the antibody is not able to reach the antigen. Again, antigen retrieval can eliminate this net and restore antigen presentation. It is crucial that before attempting any validation, a full literature review of your target is done. This will give you a picture of when and where expression is to be expected. If your antibody is a receptor, for example, you expect it to be expressed on the membrane, while if your target is a transcription factor, you expect it to be found in the nuclei. When post-translational modification or splice variant has been described, this information can be used to predict detection of multiple bind by Western blotting during your antibody validation. After collecting all possible information about your target, you should carefully select your positive and negative control. This is a key point in antibody validation. Your positive control should confirm that your target antigen is expressed in your cell or tissue, when your negative control should consist of tissue or cell where your target protein is known to be absent. You should always use several positive and negative control at the same time and confirm the expression of your target using more than one assay. If possible, you should test the expression level of your target in your sample using an antibody-independent method. For example, correlate with mRNA level using in situ hybridization or real-time qPCR. But be careful. All-through transcript data can be useful to assess the likely distribution of your protein. It should be remembered that post-transcriptional regulation do not guarantee that there will be target protein actually expressed in cell and tissue. So currently, data from proteomic studies can be useful to identify tissues expressing your antigen. So now let's have a look at the different type of control that you can use. The more accessible control are cell line and tissue, but you can also use transfected cells that overexpress your protein of interest. Another type of control can be cell line or tissue that express different level of your target, for example, before and after activation. But the really powerful tool are cell lines or tissue where your target has been downregulated or eliminated by genetic approaches, like regulated in case of a small interfering RNA and knockout in case of knockout mice and CRISPR-Cas9 technology. Let's have a look of some example of validation. Transfected cells are a valuable tool, especially when you need to check possible antibody cross-reactivity. In this example, we are validating our PIN2 antibody. PIN proteins, PIN1, PIN2, and PIN3, have a really high homology between each other. We are talking about 80%. If you do not know the epitope that your antibody is recognizing, the possibility that the antibody will cross-react is really high. For this, the reactivity of the antibody was checked by immunohistochemistry using paraffin pellet of transfected cell expressing the three family members, PIN1, PIN2, and PIN3. As you can see here in brown, we have a strong staining only in the PIN2 cell extract. The same results was confirmed by Western blotting using PIN1, PIN2, and PIN3 protein, and the same extract used for immunohistochemistry. PIN1 gives you detect a 36 kilodalton band only in PIN2 protein and HEC PIN2 cell extract. Importantly, please keep in mind that when you use purified protein or transfected cell in your experiment, you have a high amount of your target compared with endogenous expression. So before using them as a positive control in your experiment, you should first titrate the extract to find the correct concentration in which your target is present at similar level to the endogenous one. Also, another important point is that to check that the cell that you have used in your transfection, that are generally HEC 293 T cells of COS cells, do not endogenously express your target. So be really careful, because the fact that the antibody recognizes the target antigen in transfected cell does not guarantee reactivity with endogenous protein. So using transfected cell is useful, but not sufficient to demonstrate antibody specificity. So the use of positive negative control tissue with endogenous expression of your target is essential for the full validation of the antibody and the assessment of non-specific binding. This I'm showing now is an example of validation by Western blotting of our CSF1R antibody that recognizes a receptor that is expressed on macrophages. As you can see here, we use several samples, a part of a cell line overexpressing our target and an unrelated protein. We use tonsil since we know that contain high number of macrophages, and testicle as a negative control. We use 3 cases of Hodgkin disease and 5 cell line of Hodgkin disease. We also use a commercially available antibody since testing several different antibodies in parallel and observing any common pattern can be significantly strengthen confidence in the validation data that we generate. As you can see here, the result, we have a specific band in HEC cell overexpressing in tonsil and in the Hodgkin cases. These results were then confirmed using immunohistochemistry and paraffin tissue. In case of tonsil, as you can see here, you have this strong staining in brown in the macrophages in tonsil, while the testicle was negative. And in case of the Hodgkin case, you can see strong staining in these macrophages surrounding the tumoral cells, while the Hodgkin cell line was negative. If you need to detect the endogenous expression of your target on paraffin-embedded tissue by immunohistochemistry, the use of small tissue arrays can be really extremely useful. The array can contain several different positive and negative control, such as human reactive or tumoral tissue block, like in this case, or cell lines, blocks, or even pellet of your transfected cells. Importantly, after you have validated your antibody, and if you need to use your antibody to evaluate a large series of tumor tissues, you can add the small tissue array here at the bottom of the slide and use it as a control. This will increase the reproducibility of your results since you will be able to compare the result obtained in each experiment. As I mentioned to you before, testing antibody performance against genetically modified samples is a reliable way to confirm antibody specificity. This can be done using a variety of methods, including mouse knockout models. In this example, we were validating a monoclonal antibody against mouse nerve growth factor receptor. We used several tissue samples of a mouse and wild-type mouse, where you can see here the staining in liver and lymph node in the dendritic cell is this red staining. In case of the mouse where our target was eliminated by transgenic engineering, the staining is disappeared. We use also other techniques like immunofluorescence, and the same results was obtained by Western blotting using cell extract of the wild-type lymph node and the knockout lymph node. As you can see here, the band disappeared in the knockout sample. The use of knockout mice the use as a control is applicable only if your antibody recognizes a mouse molecule, so only for a small amount of antibody. But another option is to knock down the protein of interest with small interfering RNA and look for the concomitant reduction of the protein in your sample. In this example, the protein level of human MNS21 was checked and reduce using small interfering molecules. As you can see here, the protein level reduction compared with the wild type correlates with substantial reduction of antibody staining by Western blotting and also immunofluorescence. In case of the immunofluorescence, the reduction is not really significant and sometimes can make difficult to interpret the results. Also, this experiment sometimes can be difficult to optimize and often requires several more interfering RNA sequences. But thanks to the use of genome editing technique, it's now possible to generate and knock out cell lines with complete loss of the protein expression. In these examples, where we validating our tox antibody, we were using a wild-type lymphoma cell line called MOLD4, and you can see express high level of tox. When we use the same cell line that was where the tox gene was inactivated using CRISPR-Cas9 technology, and as you can see, the band disappeared. The same result were obtained using cytospins of the wild-type mold cell lines. As you can see, a strong staining here in the nuclei, the staining that disappeared in the cytospin of the knockout mold4 cell lines. All through CRISPR gene editing technology is relatively simple. The production of clonal cell line in-house can demand significant time and resources. And also these technologies cannot be used if the gene is essential for the cells. Also, a frequently used method to validate monoclonal and polyclonal antibody is the pre-absorption test. In this example, we were using our MNDA antibody that is highly expressed, as you can see here, by immunohistochemistry and Western blotting in tonsil. These are the dark dots here, and the band by Western blotting. If you incubate your antibody with an excess of antigen, the antibody will not bind to the antigen and will not recognize the antigen. So you will not see staining by immunohistochemistry and no band by Western blotting. But blocking reactivity with an excess of antigen demonstrates that the reactivity is produced by an antibody that recognizing that antigen. However, it does not demonstrate that the antibody is specific only to that antigen. In fact, because this control is often carried out using a very high concentration of blocking antigen, low affinity as well as high affinity antibody in a preparation will be blocked. So all through this control can sometimes be useful, do not provide specificity, and I'm suggesting not to use this methodology. So just in summary, during your antibody validation, please use as much control as you can. Use more than one antibody against your target, if available. Use more than one technique, and also confirm that the result obtained fit how is described in the literature. And really important, the validation data that you obtain should be available to scientific community, so you should add it in your publication. And thank you very much for your attention.
Thank you very much indeed, Giovanna. That's a sort of a tremendous overview of how to use positive and negative controls correctly. And of course, your team in the CNIO have developed, is it 150 monoclonals over the years, where you've employed all of these positive and negative controls to validate the antibodies. So, yes. As you've heard, you may have a good antibody, but if you don't have an appropriate positive control and an appropriate negative control, you'll never be able to show whether it's good or not. In the next episode, we will examine two of the most commonly used antibody validation techniques in detail, the Western blot and immunohistochemistry. And we will see that unless we are extremely cautious, even everyday techniques can really lead us astray in the most surprising manner. This is the Antibody Society webcast series on antibody validation. Thank you very much for joining us, and bye-bye until next time. The Antibody Society webcast series on antibody validation would be impossible without the generous financial support of our corporate sponsors. If you have a question about fit-for-purpose positive and negative controls for Giovanna Roncador, simply type it in now at the Question and Answers tab.